Thanks, Ada, for inviting me to talk about this topic. So is pulsed LED illumination less damaging to living cells? This is a project we've been working on for the better part of a year, and I hope you'll agree that we have some very interesting results in this to answer this question. So what I'm going to talk about today, I'm first going to spend a bit of time reviewing the current literature. So what does the literature say about pulsed illumination for live cell imaging? Then I'll present a new assay we've developed for directly measuring phototoxicity in live cell imaging. Next I'll present the results of our investigations using the pulsed LED, and I'll wrap up with a couple of things that we still want to do in the future. Okay, so first of all, what does the literature say about whether there should be an advantage for pulsed LED illumination? There's a couple of papers out there that talk specifically about pulsed LED illumination and talk about how they reduce phototoxicity. So here's a paper where they look at pulse versus continuous illumination while they're illuminating these living sperm cells. And you can see on the left, under pulse illumination, they continue to live and wiggle around. Whereas on the right, they very quickly die under the continuous illumination. So these researchers conclude that under continuous illumination, with the mercury lamp, sperm motility declined gradually, and they conclude that LED-based illumination greatly diminishes phototoxicity. I want to point out, though, that they literally used continuous illumination. So even though their exposure time was only 100 millisecond, they imaged once per second, but they didn't bother shuttering the lamp like we would normally do in our facility for all of our live cell studies. So they were really comparing 1 second illumination per image to 10 millisecond illumination. So you have a 100-fold difference in the overall exposure time. And somewhere in the middle of the paper, they admit that this is most likely accounting for the big difference between the cells living or dying. So this doesn't really say too much about whether pulsed LED illumination is advantageous when you're properly shuttering your exposure. So then there's a newer paper from just last year where they're loading their cells not with your standard fluorescent probe, but with a photosensitizer, hypericin. This is a molecule that is particularly light sensitive. When you hit it with light, it's meant to kill cells. So you can use this in photodynamic therapy. This is a sort of light-activated chemotherapy drug. So this sort of hypersensitizes the cells to illumination with light. And so definitely with this kind of molecule, you would need to limit your exposure. And they show that over a 5-hour period in continuous illumination, the fluorescence disappears immediately, and under pulsed illumination, it does not. When you use transmitted light illumination, in their study, they showed that a good percentage of the cells still divide and almost none undergo apoptosis. With pulsed LED illumination, you still have some toxicity, so not as many of the cells divide, and there is some apoptosis. But with continuous illumination, all the cells die, basically. But I would say this is even worse than the previous study in terms of the discrepancy between illumination. So they literally never shutter the exposure. So for 5 hours, they just have continuous illumination. In my view, you would never do live cell imaging this way. Whereas with pulsed illumination, the total illumination over the 5-hour period was under a second. So it's not very surprising that that the cells completely die when you expose them for 5 hours of continuous light. I don't think we've really learned much from these two papers, except that for live cell imaging, it's very important to minimize your illumination dose, and that the dose is the product of how much of the power and the duration of your exposure. So I think this is already well known, and in the AOMF, we've helped hundreds of users over the years do live cell imaging where we do exactly that. We limit the dose. So we use shutters and short exposure times and just illuminate as little as possible. And they didn't really do that in these two papers. So I wouldn't say we learned too much about pulsed LED illumination from these two papers. If we switch fields a little bit and don't look specifically at pulsed LED illumination, but just pulsed illumination in general, there's a couple of papers that I want to point out that are a little more helpful. So this paper by Rolf Borlinghaus is in relation to a kind of a confocal microscope. It's a resonance scanning confocal. And with a resonance scanning confocal, you dwell for a very, very short time on each pixel. So you have a kind of a pulsing effect for the illumination. And this is only a theoretical paper. He doesn't present any data, but anecdotally, with resonance scanners, Borlinghaus and others have noticed that there is a decrease in photobleaching and phototoxicity. So if you look at this, so he describes a mechanism by which this might take place. You have so this is a Jablonski energy diagram for a molecule under fluorescence. So when you hit the molecule with light, it goes to an excited state, and within nanoseconds, it will decay back down to the ground state and give off that fluorescence. And this is what you really want for your fluorescence molecule. But what can happen is that light can cross over to a triplet state. And this triplet state is much more longer lived, on the order of microseconds. And when the molecule eventually relaxes down, it does not give off fluorescence. So this is a dark state. While it's in this dark state, moreover, if you hit it with more excitation light, you can excite it to an excited triplet state. And both of these states are not very stable and can lead to photobleaching. So what he showed theoretically is just he did some simulations of the rate constants between all these steps, and he showed that when you have one single dose of light, that you have an initial large increase in fluorescence, but then as molecules move into this dark triplet state, you get less and less fluorescence being generated. But if you split that same dose of light into two pulses, the total area under the curves here, so that's the total amount of fluorescence, increases by about 25%, just by splitting the dose up. And the reason is that when you give it a chance to relax, now all the molecules are available to come back up to the regular singlet state that you want it to be in. So now this is for a resonance scanning confocal, where basically each pixel you dwell for a very short time, but then you give it quite a long rest, about a 100 microsecond rest before pulsing it again. And he makes the claim that many fluorophores have triplet relaxation times that match this 100 microsecond pulsing quite nicely. Now, disappointingly, he didn't show any data. So this is great modeling work. For data, we turn to another paper by Stefan Hell's group. Stefan Hell is quite well known for his super resolution techniques. These techniques use very high power lasers, and he does have a problem with photobleaching for many of these techniques. So he has to choose his fluorophores very carefully, and he's also very interested in trying to minimize photobleaching, or taking these super resolution techniques to live cell imaging, he'll need to reduce phototoxicity. So he did a study where he showed that when he normally uses his lasers, they pulse at a very rapid rate, so it's almost equivalent to continuous illumination. But he found when he slowed down the pulses more and more, he actually got brighter and brighter signals. So if he made the delay longer and longer, down to about, I think this is a 2 microsecond delay, he got about a 20-fold improvement in fluorescence and he gives the same explanation, that the reason is that he's allowing this dark state to relax in between pulses. So he draws the same kind of diagram. He talks about this lifetime being on the order of microseconds. So he says that a 2 microsecond pulse duration is, or space between the pulses is appropriate, 1 or 2 microseconds. And here's an actual result, and he's got lots of results in here that really show an advantage to the pulse, to having a break between these pulses. So you get a much higher fluorescence yield. So I did a bit of digging into what are some real triplet state lifetimes, and for a molecule like Psi5, the triplet state is on the order of 60 microseconds, and if you modify, there's some work into making fluorophores more photostable by modifying them, and the way they know they're more photostable is if this triplet lifetime is shorter. So FTSE and rhodamine, this is from years ago, that it was measured, decades ago in fact, measured to be on the order of a couple of microseconds, and more recently GFP can be, over here, about between 5 and 20 microseconds, depending on how much power you hit the sample with. So in addition to our previous conclusions that, okay, for live cell imaging, minimize your illumination dose, I think the literature quite clearly says that pulse illumination increases the fluorescence yield, at least for certain molecules, and hence reduces photobleaching and phototoxicity. It does this by allowing the dark triplet state to relax, and the off time of the pulse should somehow match this triplet state lifetime, so somewhere on the order of 1 to 100 microseconds. Okay, so in light of all that, we wanted to develop, we wanted to measure whether pulsed LED illumination is less damaging to living cells, so specifically pulsed LED illumination, and we wanted to directly measure the phototoxicity in living cells and see whether we could see a difference. So our first goal was to measure the phototoxicity directly, do this under normal live cell imaging conditions, so not crazy long exposure times, not strange photosensitized cells, but just what are most people doing, at least from what I see in my facility, for real live cell imaging experiments. And then to meet these conditions, we probably need to do something more sensitive than just counting dead cells. When I help people do live cell imaging, we don't end the experiment with all the cells being apoptotic. So we're not looking at an assay that just counts live and dead cells. We're going to need something more sensitive than that. There are these live dead cell assays, and we considered doing this, and we may still try to follow up with a comparison with this kind of technique. But in this live dead cell assay, the green dye labels live cells, and the red dye gets into cells that are apoptotic. They have a compromised cell membrane. And the way they generated this picture is they killed half the cells with ethanol and then mixed them together in the same dish. And of course, you can see a wonderful difference between them. But we did not expect this to make much sense for us when we know that we can be very careful with live cell imaging in the first place. So this is the technique we developed. We grew some epithelial cells in cover glass chambers and labeled them with 2 probes. So we labeled them first with MitoTracker Deep Red. This is our target. So this this is the probe that you would normally be using for your live cell imaging. So this could be a cell tracker or a GFP or whatever it is you're wanting to track over time in your live cells. So this is our target. And then the second probe that we put in here is called MitoSox Red. I apologize that the names both start with mito. I realize that makes it a bit confusing. But the MitoSox Red is a direct phototoxicity indicator. It's dark until it is oxidized by reactive oxygen species that are that are produced when you have toxic effects in the cells. So this is a direct phototoxicity indicator, and so with these two probes, we hope to generate phototoxicity by imaging the mito tracker, but then monitor the phototoxicity with the MitoSox. So I hope that makes sense. And unfortunately, these are both, well, they're red and deep red. So I'm just going to be coloring them red and green so that you notice the difference between them. Okay, so we imaged these on a motorized microscope. It's equipped with an incubator. It's got a definite focus to track the focus over time. It's controlled with Zeiss's Zen software. And this was important to have a fully motorized microscope so that we could do control and experimental conditions at the same time. This is always an important part of doing quantitative live cell imaging. And of course, we used the Excite XLED1 light source. This was running either in their pulsed live cell mode, set to 30 microsecond on-off times, or it was run just in continuous mode. So LED illumination in both cases, but either pulsed or continuous. Yeah, this 30 microseconds was chosen to be somewhat intermediate between that range of different triplet lifetimes that we talked about a minute ago. So this is how we did the experiment. We had a 2-well chamber. We logged 16 positions with the software, so 16 XYZ coordinates with the motorized microscope. And then we took an initial image of the phototoxicity indicator, the MitoSox, for all 16 locations. So initially, this would be more or less dark. And in order to see it, in order to make sure that we were in focus and everything, we used a bright field image to focus and find fields of view that had lots of cells. So then we set up a 5-minute time lapse where the motorized stage moved to just the 8 positions on one side of the well and took an image in each of these fields. It took a 200 millisecond exposure time of our mito tracker, the mito tracker deep red, and it went around those 8 positions about 12 times, well, exactly 12 times in that 5-minute period. So that gives us a certain dose of light for these cells, but these cells were not illuminated. And then every 5 minutes we paused and went back and measured that phototoxicity indicator again, the MitoSox, for all 16 positions. And we continued this for 40 minutes. So let's see what happened. Well, this is what the mito tracker deep red looks like. I hope you can see these movies on your screen. So what you should be seeing is that at the beginning of the movie, when this says 0 minutes, the fluorescence is brighter and it slowly photo bleaches. The cells are not rounding up and dying, so we're using a pretty gentle exposure, 200 milliseconds. We're imaging them in this 40-minute period. 12 images every 5 minutes amounts to 96 images over the whole experiment. So I realize that 40 minutes is not a terribly long live cell imaging experiment, but this would be equivalent to imaging 1 image every 15 minutes for 24 hours. So I think the point is that we're taking 100 images over the course of the experiment. And you can spread those 100 images out over whatever duration you like. I think it's quite fair to extrapolate this to longer time points, as long as you don't illuminate too often. So then I think you get equivalent results. So we pulsed. This was, I think, the pulsed illumination, and the continuous illumination gives a little more photo bleaching. I want to stress that we use the same 200 millisecond exposure time for both pulsed and continuous illumination. So there is a bit of a difference in the dose between the two, but I'll come back to that in a few minutes. So this is what the MitoSox images looks like. So this is our phototoxicity indicator. So remember, this is just one image that we take we just pause every 5 minutes, and we take one image of the MitoSox for each of these positions. And on the left, you see one of the control fields of view where we didn't illuminate the MitoTracker, and on the right, you see one of the positions where we used pulsed illumination of MitoTracker. And you can see that there's quite a bit more activation of this MitoSox dye, this phototoxicity indicator, when you illuminate the MitoTracker. So that was encouraging. Then we turned off the pulsing, turned it in, so it turns it into a regular continuous LED light, and you see that it's dramatically brighter MitoSox signal compared to the control. Okay, I'll let that go through one more time. So, there definitely is more activation of this MitoSox phototoxicity indicator when you use continuous illumination. When I go on to, so here I've put pulsed and continuous side by side. When you put them side by side, this is even more apparent that the continuous illumination gives you more phototoxicity. And just so you know that I didn't take the best field of view to show you, I took a typical field of view. Here are all 16 wells tiled together in a mosaic for you to peruse, and you see the ones, there's a bit of variation from field to field, but in general, on the right-hand side with continuous illumination, there's much brighter expression compared to pulse illumination. There's no doubt about it. Okay, so this is an exciting result. Let me show you some analysis now from these slides. So we used image J and did pretty standard things. We removed some of the noise. We're trying to minimize our illumination so we don't have terribly high signal to noise in these images. So we did a Gaussian blur to remove some noise, subtracted the background by choosing a blank area where there was no cells, set a threshold in an unbiased way based on an auto threshold that looks at the stack histogram, and use that to measure the integrated optical density, which is the percent area stained times the intensity of that staining. And here's the result. So there is a base increase in the MitoSox signal when you don't illuminate the MitoTracker at all. That's not too surprising because there are reactive oxygen species always being produced in cells for various reasons. It's hard to know whether this indicates the MitoSox itself may be slightly phototoxic to cells. We're not sure about that. But there's that base level. With the pulsed, you see that you have substantial increase over the control level and even more of an increase with continuous. Okay, so again, I want to point out that I don't want to make the same mistake as those first two papers that I showed you where they had very different illumination times. So I want to be fair about this. We have 200 millisecond exposure times for both the continuous and the pulsed illumination. And that's not quite fair because with the pulsed illumination, the lamp is off half the time. It's a 50% duty cycle. So I'm going to correct for that in the next slide. I'll show you that in a second. So there's no doubt that the MitoSox phototoxicity indicator decreased for pulse illumination because there was a lower dose. But if you look at this graph, you see that the distance between control and pulsed is less than half the distance from control to the continuous illumination. So if we look at the relative increase above the control line, if we look at the relative increase and correct for that for that control level, and then I'll double the pulsed values, so that now that should account for having half of the dose with pulsed. And you see that there still is a residual advantage for pulse versus continuous. So remember that lower mitosoc signal means less phototoxicity. So with the pulsed, even if we double the values that we had for the pulsed illumination, we're still about 25% below the mitosoc signals that we see for continuous illumination. Okay, so I think this is a very exciting result. First of all, I was very pleased with how well this mitosox worked as a sensitive live cell imaging assay for phototoxicity, for measuring phototoxicity. Mitosox was really meant for toxicity studies, and to my knowledge, it's not been applied specifically for phototoxicity anywhere else. So I think this is the first time that we're using it this way, as far as I know. And the other thing that's quite exciting is that this is such a sensitive assay that we were able to see quite clearly a difference between pulsed LED illumination and continuous illumination. And the pulsed LED illumination comes out well ahead of the continuous illumination in terms of reducing phototoxicity. So part of it is definitely because the pulsing leaves the lamp off half the time. Well, this actually satisfies the very first principle of doing live cell imaging, which is just simply reducing your dose. But in addition, there seems to be a real advantage that the pulsing itself gives an additional reduction in phototoxicity. So there's still some work to be done. I'm aware that it would be nicer if we had equalized the light dose in the first place by cutting down the LED power for continuous illumination, or doubling the exposure time for pulsed illumination. So just equalizing the light dose. And this is a study that we're working on now. But I believe it will still show this 25% advantage for the pulsed illumination. I would also love to extend these experiments. I think these results I think that you are able to extrapolate this to hours-long experiments, provided you spread your illumination dose over those hours. I'm not sure, like when I see the base level of mitosocs increasing, even when we don't illuminate the cells, I'm not sure that the mitosocs assay can be extended specifically to 24-hour experiments, but we can definitely extend it by several hours, and we're working on this as well. And then I think it's fair to say that we should probably try to compare this to more of a gold standard for toxicity studies and try to do a live dead cell assay, but we're definitely going to have to increase our light dose for this, and we probably won't be in a regime that we would normally use for live cell imaging. But I'd like to do that as well. So on that note, I'd like to I want to particularly acknowledge Feng Shu, who works with me at the AOMF. He's the one who has been doing all the countless hours of optimization, live cell image. He did most of the live cell imaging experiments. And so he did a lot of work for this project, so I thank him very much. And then Kavita for scientific direction from Lumen Dynamics. So thank you very much.